Hello, friends. Very good morning to everybody. As we are here to learn the gene interaction, in that we saw that gene interact by two ways, that is, allelic interaction and non allelic gene interaction. Well, in allelic gene interaction, we learned the incomplete dominant example of a mirabilis jalapa, and in that incomplete dominant and codominance. So what we saw in incomplete dominance, we saw that regarding the species of Mirabilis jalapa, it was crossed with the red flower into white, and what it got in F1 generation, that is pink one, that is completely different character. The genes has played complete different role here, and they delivered the pink color in the organisms. Well, on self-fertilization of this pink flower plant of Mirabilis jalapa, what it produced in a F2 generation, in next generation, that was the red as well as the white has reappeared, and two pink was found in it. That is why the ratio was 1 is to 2 is. So this was what regarding the incomplete dominance, that it got the ratio, phenotypic ratio and genotypic ratio, that is 1 is to 2 is to 1, that is both genotype as well as phenotype are equal in the case of the allelic interaction in incomplete dominance. Now we move towards the codominance. What is the exact definition, or what we can say that the term is used for the phenomenon? Why this term is used, that is codominance? What we get in codominance? Well, in codominance, most of the example we see, that is the color coat of the cattle. Now we are aware of the cattle, what we see in the cows and all this. This is a condition in which both alleles of a gene pair in a heterozygous condition are expressed fully, with neither one being dominant, neither one being recessive to the other. What we see in monohybrid, here we see completely different. What we see in incomplete dominance and in the codominance. The best example of this, that the color coat of a cattle. Well, when we select a phenotype color coat of a cattle, that is the red cattle and the white cattle, and when we look out at the genes of this red cattle, that are the pair of alleles responsible for this color, and those are capital R, whereas in a white cattle, though it is a recessive one, but it is the capital W, nor we see here smaller letter. But what will happen in F1 generation, that R1 as well as the W1, when they are crushed in the cell division, The characters shows somewhat different in F1 generation, that is the Roan one. The new we got it there. What we got there? New one, the Roan one. As we saw in incomplete dominance that of Miravilla Jalappa, that from red flower as well as the white flower we got pink one. Same way what we saw in the codominant, that was new color appear, that is the Roan one. But what? F1 generation undergoes self-pollination. When this F1 generation undergoes self-pollination, means roan into roan. Phenotype roan into roan. Genotype, that is RW and RW. When we put these genes or this letter in the Punnett square, what we see that? We see that we do find the ratio, that is 1 is to 2 is to 1. But what we see in the codominance in case of coat color of a cattle, that the red one has reappeared, the white one has reappeared, and the roan that we got different in the F1 generation has got 2 in number, that is the means no dominancy and no recessiveness have been observed. That is co1, somewhat equivalent dominance as well as the recessive one. Well, while studying the Mirabilis jalapa, what we see the same one, the red in case of this plant, the red color flower and the white color flower were observed, but the genotypes of these flowers were pure. Okay, we get here, that is the pure one. Whereas in the coat color of a flower, we do get the red one and the white one, that is the pure parent, and both the roan one. So friends, what we see here, that is the non-allelic gene interaction. The second one, in the case of gene interaction, we have seen the allelic gene interaction, and now we move towards the next concept of the gene interaction, that is the non-allelic gene interaction. What we see in interallelic gene interaction, the independent genes located on the same or on different chromosomes interact with one another for the expression of single phenotypic character of an organisms. Well, I repeat the phenomenon of the non-allelic gene interaction. The phenomenon is that the independent genes which are located on the same location or on a different chromosomes, these genes which are located on this same or a different chromosomes interact with each one another for the purpose of the expression of a single character of an organism. What we see here in non-allelic, we see that the genes which are located on a same location or on the different chromosomes, they interact with each other, they communicate with each other for the purpose, for the aim, expression of a single phenotypic character of an organisms. Now, in what way the interaction carries, how the interaction of the non-allelic gene takes place, there are four types. Which are those? The number one, that is complementary gene action. The number 2, that is epistatis. Epistatis is further divided into two types, that is the dominant one as well as the recessive epistatis. As we saw in the allelic gene interaction, well, incomplete dominance and in codominance, what we saw in codominance that neither the dominance, neither the recessive were expressed. What we studied the color coat of the cattle. Here in the epistrates, that the dominant epistrates and recessive epistrates can be studied. Third one, the duplicating factor. Duplication takes place. Supplementary gene action. Supplementary gene action. Well, while studying, the very first we study the complementary gene action. What is the complementation or the complementary action of the gene? What happens in this type? Two or more independent dominant gene. Look out here. Dominant gene, two or more independent dominant gene occurring in different loci or on the different location on same chromosomes or on different chromosomes interact with each other producing the character, but neither of them produce that character in the absence of other. Batson and Punnett studied the inheritance of a flower color in a sweet pea, that is Lathyrus odoratus. So, well, friends, we repeat the phenomenon of the complementary gene action, that is, two or more independent dominant gene occurring in different loci on a same chromosome or on a different chromosomes interact with each other producing the character. But what happens? Neither of them produce that character in the absence of other. Well, here you can see the complementary gene action, that is the white flower and the white flowering plants of them. Now look out at the genes. There are the two or more independent dominant gene. Here we find the two gene, that is the capital C, smaller double A in case of female one, smaller double C and capital double A. What we see in the F1 generation, that white flower produce red one. But this is the phenotype. What is the genotype? Genotype is one is the capital C with the smaller letter C, another one is the capital A with the smaller letter. What happens when we crossed this race one in F1 generation? Let them allow the self-pollination. What happens then? Phenotype. Red into red. Okay, what we get? The genotype, the genes that we separate them, we find the in the Punnett square that capital C, capital A with capital C with the smaller a. These are the two. Smaller C with the capital A, and the fourth one, that is the smaller c with the smaller a. Well, friends, look out at this genotype now. We are putting it in the Punnett square. Female one on the other hand and the male one on the other hand. While crossing over it, we came to know that the complementary gene has shown their actions. In what way? We found the 9 red one and 7 the white one. Nor the dominant nor the recessive. The 7 one whites, which were the maternal as well as the paternal one, and the red one, the number of red one was increased by 2. Now, this is what the phenotypic ratio of 9:7. When we talk about the genotype ratio, what we come to know, that red one, that is the 9. See, look out at the genotypes: capital double C and capital double A. Capital double C, then the bigger letter A, smaller a. Then look out at this third one, capital C with smaller letter and both the double A with the capital. And in this way, we got the 9:7 ratio of the complementary gene action. So friends, with this we stop our today's lecture. We'll find out rest of the part in the next video. Thank you.